Just one seemingly simple molecular modification allowed chemists to create a new version of LSD that increases the plasticity of the brain while also fixing a major shortcoming of traditional psychedelics. To add even more allure, this new substance might even be a much more potent antidepressant than ketamine. In this interdisciplinary video, we'll review some familiar concepts and biology in neuroscience, learn how this LSD analog was discovered, and check out how it performed in initial experiments. As usual on the channel, we'll also deep dive on how this molecule was synthesized and quiz you on some chemistry, including a reaction that most students have never heard of in their entire life. So what has happened recently in the field of medical controlled use of psychedelics? Well, after last year's somewhat logical FDA rejection of ecstasy as a therapy for PTSD, LSD has entered new phase 3 trials in generalized anxiety disorder and in major depressive disorder. These disorders affect literal millions of people in the US alone, so there's a lot of hope for positive data over the next few years. We've also seen new data on microdosing. Over the last years, this hot topic has been dubiously dubbed a magic bullet for improving health, personal efficiency, or even professional success. In ADHD, researchers published the first double-blind, placebo-controlled clinical trial of repeated microdosing of LSD. The dose was already on the higher end, but did not lead to significant improvements in self-rated and observer-rated ADHD measures. You can argue that 53 participants are not enough to detect small benefits, but the numeric improvements are not particularly encouraging. Funnily enough, even the majority of participants receiving placebo assumed they had been receiving LSD, and in turn, these showed high reductions in ADHD symptoms. Such expectancy biases really highlight the need for proper clinical trial conduct to tell us what really works and what doesn't. Stepping back, what is the biological reason for research into LSD and other psychedelics? One major part relates to neuroplasticity, or the ability of the brain to adapt or remodel based on its dynamic environment. This is essential for learning, memory, etc., and is highest during human development, but lower during adulthood. These are obviously very complex pathways, but simple ways to think about it are the growth of new neurons, which are the core building blocks of the brain, or simply the addition of more connections between existing neurons. As one example, dendritic spines are the tiny protrusions that serve as contact points between different neurons. A decreased density of these dendritic spines is a hallmark of several neuropsychiatric diseases. Because psychedelics can stimulate regrowth of these dendritic spines, they might be promising potential treatments in depression or mood disorders. If you're interested in the nitty-gritty science of this, you can check out this very recent review to learn more about the different pathways and current plasticity evidence across compounds. One powerful feature is that beyond short-term activation of neuroplasticity by psychedelics, we might also see long-term benefits from a single or few doses. For example, the growth processes last days, but the new structures might survive for weeks or months. Please note that drug-induced plasticity changes are not necessarily positive. As we always emphasize here, there are safety risks and issues associated with any exogenously administered substance. Another drawback of psychedelics are hallucinations. In the therapeutic setting, these can require close clinical supervision or increase anxiety or stress of patients. This isn't catastrophic and may actually be desired by some people in the recreational setting. But in schizophrenia or borderline personality disorder, hallucinogenic substances could be counterproductive or increase risks, despite them offering potential neuroplasticity benefits that could help the underlying issue. Therapeutics so far look like a double-edged sword, but I have some good news for you. It looks like even healthy adults can get neuroplastic effects from quote-unquote exercising the brain with puzzles, crosswords, and memory training. The general activity of learning anything can increase the development and survival of certain neurons. A cognitively active lifestyle also significantly reduces the risk of dementia, so we all need to be lifelong learners. Even certain types of video games might help, but don't use this as an excuse to play 16 hours of World of Warcraft a day. There is a much better way to not only exercise your brain, but simultaneously develop useful skills. The learning app Brilliant. With thousands of interactive lessons in science, math, programming and other areas, Brilliant helps you get smarter every day. Because Brilliant uses visualizations and combines different engaging formats like puzzles or quizzes, it gives you a broad cognitive activation, which we mentioned is so critical. Daily encouragements and competitive features keep you motivated and help you establish a learning routine, so you're on track to become a lifelong learner. Brilliant covers all critical disciplines to satisfy your interests and can even equip you with core skills for your career. The courses in the science category will enhance your scientific intuition, make you a better problem solver, and help you better understand our world. On the other hand, Brilliant's data science course is perfect for learners of any level to work on their data visualization and analysis skills. The lessons and interactive problems are a great way to truly exercise your brain. To try everything Brilliant has to offer for free for a full 30 days, visit brilliant.org/totalsynthesis or scan the QR code on screen. Or you can click on the link in the description. You'll also get 20% off an annual premium subscription. Let's get back to the main topic of this video, non-hallucinogenic variants of psychedelics. Essentially, we want to find something that is as promising as the natural substance, but has a better safety, tolerability, or compatibility profile. We've discussed similar things for 6-methoxy-DMT and different analogues of ibogaine, which could be safer for the cardiovascular system, for instance. In these videos, we've also seen that researchers test the trippiness of compounds by assessing the involuntary head twitch response which they generate in mice. Well, for LSD, some supposedly non-hallucinogenic variants have been discovered already, ranging from minor changes like the attachment of a bromide to a more major modification of the diethylamide substituent. The latter compound, lisuride, has been clinically studied already half a century ago and was ultimately approved in some countries to treat Parkinson's disease or migraine. As we just said, there is a good correlation between a molecule's hallucinogenic potential in humans and its ability to induce the head twitch response in mice. The more head banging, the spicier the molecule. The test isn't a perfect prediction, but it's still a good one. We can see that LSD, similar to other hallucinogenic psychedelics, increases HTR in a dose-dependent manner compared to the control. In contrast, lisuride actually decreases head twitching, and many references claim that lisuride is non-hallucinogenic at normal doses. If you're a fan of the channel, you will know better and anticipate that science is rarely black or white. Indeed, there is early evidence like this human data referenced in the funnily named journal Headache in the 70s. But it doesn't take a genius to look into the safety information of approved lisuride formulations to see that hallucinations are very common and occur in more than 10% of cases. So much for non-hallucinogenic, and there were equally old case reports that lisuride is not that innocent. Now check this out. This is why I like digging up old scientific literature. One moment you're debating whether you should double check some innocent information you read, and a moment later you're going through diligent documentation of crazy hallucinations. Feel free to pause and take a look. The last one is particularly wild. Even if lisuride is not completely non-hallucinogenic, it definitely failed the HTR test. But why, considering that the structures are so similar? For this, we have to scratch the surface of some cellular biology pathways, which we all know are very simple and very easy to remember. You surely have heard of serotonin, a critical neurotransmitter. There's a broader family of receptors which recognize serotonin, But the 2A receptor is one of the primary sources behind the plasticity benefits, but also hallucinations of psychedelics. You can imagine this like a chain reaction or a cascade of dominoes. If this receptor is activated and importantly adopts just the right 3D shape, it can affect other downstream pathways which do not only improve plasticity but also trigger hallucinations. If we look at this 2A receptor in more detail, we see that it has two distinct binding regions, a lower pocket in green here and an upper extended pocket in red. The situation is already plenty complex for the natural substrate serotonin and the magic mushroom psychedelic psilocybin. These molecules are much smaller than LSD, so they can even adopt two different binding modes, either primarily binding to the lower or the upper pocket. On the other hand, LSD and lisuride bind to the lower pocket with the tricyclic ergoline system, and their side chains reach up into the extended binding pocket, so they span across both pockets at once. This is exactly what we see when we zoom in. In the lower area, the indole nitrogen acts as a hydrogen bond donor to contact the receptor's amino acids. Both LSD and lisuride look the same here, so the difference in head twitch response must arise from minor differences in the upper extended binding pocket. This might all boil down to the engagement of a single tyrosine amino acid, which can only contact one ethyl group in lisuride compared to two ethyl groups in LSD. This different binding makes the receptor adopt a slightly different conformation, and in turn, this affects how the next dominoes in the pathway will fall. We're not going to talk about the specifics to preserve our sanity, but you get the point. When you change the structure, you change the binding and conformation, which ultimately can change the effects in terms of HTR and/or hallucinations. Remember that the 2A receptor has two binding pockets, and we just saw what happens when you alter the fit in the extended pocket. If you're thinking along, you might wonder if fiddling with the lower pocket could have an impact as well. After all of this setup, we can finally check out this latest variant of LSD. Olson and co-workers simply asked themselves how a hypothetical constitutional isomer of LSD lacking the indole would behave. We know now that the NH bond is a donor for LSD in the lower pocket of the 2A receptor, so this change is conceptually the opposite of the lisuride change, which had a different binding in the extended pocket. Computational modeling would suggest that this compound, dubbed JRT, simply moves 0.1 nanometers away from the usual hydrogen bond acceptor amino acids. It might seem like a stretch to believe that this small change can already deliver a different therapeutic profile compared to LSD, but science is all about just going for it and seeing what happens. Well, the first issue is that we have to find a way to make this new compound. The chemical synthesis of normal LSD is a solved problem. As we've seen in previous videos, the first synthesis of the LSD precursor, lysergic acid, was published by Kornfeld and Woodward and relied on an aldol condensation among many other reactions. In contrast, the very efficient 6-step synthesis by Smith used a palladium-catalyzed Heck reaction to connect the two sp2 centers. If you're into biotechnology, even genetically engineered yeast cells can make lysergic acid directly from glucose. The figure even includes a nice smiley face, so you have to love it. If dozens of LSD syntheses have been produced over 70 years, then it should be no issue to create this analog, right? This couldn't be farther from the truth, as developing the synthesis required almost 5 years. This sounds really annoying, but on the bright side, it allows us to review some organic chemistry. The retrosynthetic strategy was based on a quite straightforward disconnection, an intramolecular alkylation of our repositioned nitrogen. This indole was combined with the other half through a Suzuki cross-coupling, so this disconnection is similar to the 2023 Smith synthesis. All of this means that this functionalized pyridine is one of our first key intermediates. Our starting material you can see here is a free carboxylic acid, so the first no regret move is to create the diethylamide present in LSD. So far, so good. Remembering the pyridine, you'll realize that we need a hydroxymethylation reaction. In a dream scenario, this is just one step, but these guys had to take a 7-step detour. They didn't disclose all their synthetic attempts, but probably shorter approaches did not work because the electron-deficient ring was either not reactive enough or because selectivity was an issue. The logic here for this indirect approach was to first introduce a halide group and then later form the carbon-carbon bond. Importantly, the process here uses the more reactive oxidized pyridine N-oxide as an intermediate. It's not sorcery, but let's still check out this chlorination mechanism in more detail. The oxalyl chloride serves a dual role here. First up, the N-oxide is acylated and the released chloride anion can add to the electrophilic pyridine, similar to what we know from nucleophilic aromatic substitutions. This reaction is pretty selective for the position alpha to the bromide, which seems to be more activated. This intermediate has lost its aromaticity, but a final deprotonation can occur, kicking out the acyl group, which fragments further. Keep in mind for later that this is a classic leaving group, also a so-called electrofuge, as it takes the electrons in the NO bond with it upon departure. Next up, instead of directly creating a CC bond, the chemists first exchanged the chloride for a more reactive iodide. This is some more pyridine chemistry, but with a nice twist. The reaction uses the electrophilic TMS chloride as a reagent, which can react with the nitrogen lone pair, just like oxalyl chloride earlier. This again increases the electrophilicity of the carbon next to it, so the iodide adds up in there. The system now has two choices. It can immediately reverse the addition by kicking out the iodide, or it can kick out the chloride. All of these steps are reversible, but the exchange reaction is still surprisingly efficient. This is because the TMS chloride byproduct has a much lower boiling point than TMS iodide, so it can be removed from the reaction equilibrium by heating and trapping the TMS chloride with a Dean-Stark apparatus. The twist is that this silyl group is a nucleofuge and not an electrofuge like the previous leaving group. With the more reactive iodide in place, the chemists performed a cross-coupling reaction with diethylmalonate as a 1-carbon equivalent. The esters were not needed, so they were hydrolyzed and decarboxylated. The first decarboxylation is pretty easy because the resulting anion can be stabilized by the remaining group. In contrast, the second one required acidification and prolonged heating. Alright, after this painful introduction of the methyl group, we can oxidize it to the alcohol. Can you figure out how this works? This is actually a mysterious name reaction, but I doubt many know its name. Feel free to comment below, but no cheating. The reaction is really similar to our first chlorination with oxalyl chloride. In hopefully no surprise to anyone, the oxidation gives the N-oxide. Next, nucleophilic attack of the anhydride leaves an acyl group at the N-oxide, converting it again into a nice leaving group. The nuance here is that due to the presence of the methyl hydrogen, we can temporarily disrupt the aromaticity to give an exo double bond. This intermediate can rearrange, exchanging the rather weak NO bond for an energetically much more stable constitution. The carbonyl group transformed into an alkyl ester, which we can simply hydrolyze with base and water. Such sigmatropic rearrangements might look random, but they do play a part in different heterocycle syntheses, such as the classic Fischer-Indoll synthesis or this very recent example from the well-known Professor Kürti and co-workers. In this method, N-oxides are attacked by hydroxylamine in presence of anhydride activating agents. The mechanism isn't shown here, but it's similar to the chlorination with oxalyl chloride that we saw. The hydroxylamines aren't just great nucleophiles, but because they come with vinyl substituents, the products can undergo free rearrangements. We again swap out the weak NO bond for stronger double bonds, and ultimately get some nice 7 ASA indoles. Let's wrap up our fancy LSD synthesis. With the functionalized pyridine in hand, the remainder of the approach is pretty straightforward. An alkylation incorporated the required methyl group and primed the system for reduction with 3 equivalents of sodium cyanoborohydride. This reduced 2 carbons with preference for the thermodynamically more stable anti-diastereomer. The remaining double bond is needed in our final product, and it allows another cross-coupling reaction to incorporate the indole ring. The indole is obviously nucleophilic, so it gladly cyclizes after conversion of the free alcohol to a leaving group. Because we didn't use chiral starting materials or any enantioselective steps, our product is a mixture of enantiomers. We often highlight that structure defines the biological activity. In our last video on ibogaine, we've seen that the unnatural isomer of ibogaine has a lower activity. In this case, we can use chiral chromatography to simply isolate the 2 enantiomers and test them separately. This begs the question, how do they interact at biological targets? Unsurprisingly, the 2 enantiomers had different engagement and potency. Plus, JRT was active at even more receptors and had stronger affinities than minus JRT, so it looks more promising. Unlike LSD, both enantiomers were not particularly active at other targets, like dopamine, histamine, or adrenergic receptors. This selectivity might be beneficial because targeting more receptors than truly needed is a recipe for unintended side effects. They were all active at the critical serotonin 2A receptor though. Now comes the critical question. Does the different binding of JRT, just like lisuride, impact the downstream dominoes of the 2A receptor activation, ultimately reducing the hallucinogenic potential? The answer is yes, because JRT did not induce head twitch response in mice. Numerically, it looks like higher concentrations led to some head twitches, but this was not statistically significant and definitely lower than the massive responses seen for LSD. This is great news, but we've seen with lisuride that the head twitch response assay is not a perfect prediction. So it could be that JRT ends up being hallucinogenic anyways. The main question is if the molecule still improves neuroplasticity, the thing we're actually after for the therapeutic benefit. This first data is from in vitro experiments performed in microplates in the lab. This first view shows the effects on dendrites in neurons of rat embryos. These extensions that look like tree branches are critical for the connections between neurons. Qualitatively on the left and quantitatively on the right, we see that LSD and our isomer JRT increased the formation and complexity of dendrites. The gray bar is a negative vehicle control and the purple bar is a positive control. This is clozapine, an antipsychotic medication used in the treatment of schizophrenia, so one of the conditions where psychedelics would be very promising if they weren't hallucinogenic. Clozapine also seems to decently increase dendritogenesis, but with many side effects, it's far from a perfect drug and there is a big need for new alternatives. JRT also looks promising when zooming into the dendrites and looking at the tiny spines which form contact points or synapses. JRT promotes the growth of these dendritic spines, adding more indicators that it could improve neuroplasticity through different mechanisms. Another set of good news on the safety side came from in vivo tests. One of these was a forced swim test, an assay which can predict antidepressant potential. This test characterizes the behavior of rats when they're injected with a substance and placed in water which they cannot escape. Antidepressant drugs lead to a reduction of immobility and an increase in active behavior. The positive control in this case was the anesthetic and antidepressant ketamine. Not only is JRT also efficacious, but it looks to be 100 times or more potent than ketamine based on the concentrations. They performed other tests, but to not overdo it with the animal experiments, we will wrap things up here. In summary, we saw that this new compound is non or at least less hallucinogenic that it might increase neuroplasticity like LSD, if not better, and that it also could have antidepressant effects. By the way, it also doesn't seem to inhibit hERG ion channels, which is one of the issues of the psychedelic ibogaine. We can only say more about JRT and all the other recent psychedelic-derived drug candidates if someone actually starts to prepare and initiate clinical trials in humans. Until someone does, we'll continue to use these preclinical case studies to learn something new about science and chemistry. If you found this video interesting, maybe share it with a friend of yours and make sure to check out the channel. Thank you for watching, and a big thanks to all my channel members. As always, I hope you will join me again in the next one.